No worries, no worries. So takes as long as it takes to sort out.
I just need to restart my PowerPoint as well. Try sharing.
We're blessed so far with when it came to technical problems, so it was bound to happen one day.
Yeah. How about now?
Same thing. I can see that you're sharing the screen, but I can't see any slides. And I think you're frozen again. Yeah, dropped out.
It just drops me out every time I try to share my screen, which is a bit of an issue.
Just a little bit. What we can try is you could send me your presentation and I share it. And then you just give me a nod every time you need the next slide. Oh, I think we lost her again. Very unstable connection. Apologies to the ones who joined us just now. We are having some technical issues at the moment that we're trying to sort out. And hopefully the talk can start very soon. I've never seen that when someone tries to share a presentation, they get kicked out of Zoom. I can see some of you sending suggestions in the chat. Thank you. I tell her when she's back. Well, in the meantime, let me use that time to introduce our speaker so we can go straight into the presentation when it works. So Angelika is presenting today the finding the network balance in Parkinson's hallucinations. She currently works 60-40 at the moment, I think, research and clinical as the Alzheimer's Research UK Clinical Fellow at UCL Queen Square, the Institute of Neurology. And she graduated from the Athens Medical School. Oh, right. Okay. So just got to pop up that someone activated closed caption. Well done. And she, as I said, is working clinically and on the research side, and her research is based at UCL, and her clinical work is based at St. George's in South London. Now, when she comes back, hopefully, the presentation will work and we can dive right into it. I do apologize for the delay. It's the first time we're actually having technical issues. Great, and she just shared her presentation. So worst case scenario, we can always do a PowerPoint karaoke where we look at the slides and tell the story that she was supposed to tell us. Okay. So let me find the presentation on my computer. She's back in the room. Trying to connect. Here we are. Hey, I got your presentation. So if it doesn't work, I happily share your slides.
So it just restarted again. So Zoom doesn't like me today. And it was fine when we were at the start.
It's always the case. The second you hit the live button, things go wrong. And before everything is fine.
Okay, should we try it once more?
Try it the last time, and if it doesn't work, then I share your
Yeah.
presentation and you just let me know when you need the next slide.
Yep. Okie dokie.
And don't get stressed by this. It's all fine.
That's all right.
Just always happens when you hit live, things tend to go different than planned.
Okay, I'll try and do this a different way. It doesn't look particularly promising. Does it?
No, it's the same thing.
Okay. Yeah.
So
and now it's gonna crash me out again. So
let's see if I can override it. She's still here. No, we lost her. Okay, I'm just going to wait for her to be back in the room before sharing the slides. And we're going to have a proper academic quarter of delay for today's talk.
Can't even stop sharing.
Okay, here we go. Okay. You're still frozen, aren't you, Angelika? Yeah. Okay, she's come back in. And then we're gonna try a new way. Oh, wow, I see you twice on my screen now. Okay, you don't touch anything.
I'm not touching anything.
Don't touch anything. I'm sharing your presentation.
Okay, all right.
Here we go. Is that working? Can everyone see it?
Great. I can see it, yeah.
I used the time you weren't with us to already introduce you, so
perfect.
Take it away.
Okay, thank you. Apologies everyone for this. So I'm Angelika. I'm a clinical postdoctoral clinical research fellow at UCL, and I'm going to talk to you today about some of the work I did mostly through my PhD in the past 3 years, trying to use neuroimaging to understand hallucinations as a disorder of networks. So thank you, Stephanie. So although we mostly think about Parkinson's disease as a disorder affecting movement, it's actually neuropsychiatric symptoms, and particularly visual hallucinations, are very common, and they're very diverse in patients with Parkinson's disease, ranging from illusions to pareidolia, so seeing faces when there are no faces, to very complex, vivid visual scenes. And as I said, hallucinations are very common. Up to 70% of patients with Parkinson's disease will experience them, and they're associated with a lot of worse outcomes for patients. So worse quality of life, increased risk of subsequent dementia, higher carer burden, and now the single best predictor for nursing home placement in Parkinson's. But thank you. So far, research into trying to understand how and why they happen in the brain hasn't been as successful in using neuroimaging, at least, and partly that is because no clear single brain region is responsible. I think this meta-analysis here by Lenka in 2015 highlights that quite nicely. So they looked at various different studies using VBM, so voxel-based morphometry, so gray matter volume, and as you can hopefully appreciate, there's a lot of different spatially and functionally distinct regions that are associated with hallucinations. But in contrast, if we look outside of specific regions and look at large spatially distributed functional brain networks, there are changes in function in hallucinations, specifically the default mode network. So there's a lot of aberrant activity, but there's also increased connectivity between the default mode network and the visual cortex, for instance. So we need to kind of move away from thinking about mechanisms of hallucinations from different regions of the brain in looking at large whole brain networks. And What we also know is that vision is affected in Parkinson's quite early on. So people with Parkinson's have difficulties copying intersecting pentagons. They have changes in color vision. So they also so move on a little bit. I have a lot of animations as well, but doesn't help. So they say differences in color vision. They say changes in contrast sensitivity compared to people without Parkinson's disease. And all these visual deficits are linked with worse outcomes in people with Parkinson's. And more recently, work from several groups, as well as here at UCL from our group, has shown that there's also thinning at the back of the eyes, the retina in specific layers. And again, this is correlated with subsequent development of dementia. And even looking at the most crude and basic visual test, which is the visual acuity using the Snellen chart, people with Parkinson's perform worse than age-matched healthy controls. So the visual system, even as early as the back of the eyes, the retina is affected in Parkinson's disease. But just because you have disorder in your visual system doesn't really explain why people with Parkinson's see these very, very complex and vivid and well-formed visual hallucinations. And that is because vision is not really just a passive process, but is influenced by our prior knowledge. And I think this is a really good example of how prior knowledge influences visual perception. So if you take a moment to look at this photograph and can you think of whether you can see any objects in this photograph. So some of you may be able to see an object, but likely most of you will just see a brick wall. But if I now highlight you the object, or Stephanie does anyway, which is that cigar right in the middle, and now you see the same photograph again, the cigar is kind of really, really clear to perceive. And once you see it, you cannot unsee it. So the final visual percept is a combination of both the sensory evidence, the signal from our eyes, and our prior knowledge from memories, goals, expectations, and context, etc. And it is has become clear in recent years that in Parkinson's disease, the way that the brain accumulates sensory evidence, so bottom-up influences in perception, is affected. So I showed you already a lot of visual deficits and changes throughout the visual system. What is less clear is whether there is also change in top-down influences, so prior knowledge, and that may be potentially relevant because we know from really nice work by Teufel and others in 2015 that in hallucinations associated with psychiatric disease and healthy people who have first step to psychosis, prior knowledge is overweighted. So if the same is true for Parkinson's disease, there may be transdiagnostic implications. So, but even if we find this kind of increase in prior knowledge and there is imbalance between those computational networks, where exactly would that imbalance happen? So many studies assessing priors specifically for visual perception have implicated frontoparietal regions, so the posterior parietal cortex, medial, and also prefrontal cortex. And here's just one example, Baranev, that shows using fMRI and a behavioral paradigm, they show that the dorsolateral prefrontal cortex shows larger activation for trials where subjects had an expectations compared to trials where participants had no expectations. But although the prefrontal cortex itself may be a good candidate for generating priors, there's a lot of ways that the PFC kind of exerts its influence over a lot of other brain regions by changing functional connectivity and mediating kind of sensitivity to signals. And in all occasions, these changes in effective or functional connectivity between regions are likely to be mediated by signals through anatomical connections. And all of these regions, the prefrontal cortex, the temporal cortex, the parietal cortex, the visual cortex, are very strongly interconnected. And there's a lot of different levels where an imbalance between computational networks may happen. So it could be the prefrontal cortex itself, the thalamus, the visual cortex itself. It could be due to changes in the relative connectivity between regions rather than a disorder in the region itself, or it could be a combination of those factors. So here today, I'm going to kind of touch on kind of trying to answer this question a little bit crudely by taking through you 4 experiments. So first, I will kind of talk to you about understanding cognitive mechanisms of visual hallucinations. So the change between prior knowledge and sensory evidence. Then I will try and walk you through some longitudinal structural white and gray matter changes in patients with hallucinations compared to those without hallucinations. Then I will focus a little bit on the thalamus as a potential driver and kind of move through my animations. Finally, I will try to kind of bridge structure and function by modeling the effect that structural brain changes have on functional brain dynamics. So if we so these are going to be the 4 main things that I will talk to you today about. So if we move on, so first to try and understand computational mechanisms first. So to do this, we're specifically interested in quantifying, isolating the effect of prior knowledge. And to do this, we use these 2-tone stimuli that when you first see them, they appear as random black and white patches, but when you see the template from which they were produced, they generate a strong percept. So hopefully by looking at the image, most of you will just be able to see like a black and white patch. But if I show you the template from which it was produced, and then you see the image again, so that most of you now will be able to kind of see the outline of the baby and the water and the bath. So what we did is we used these 2-tone stimuli, and we had a lot of them that contained a person or a human anyway on the left, and then some of them that did not contain people but had animals in them. And we presented 57 kind of participants with these 2-tone stimuli and had a very simple 2 alternative force choice paradigm where we asked them whether they could see an image in the picture or not. And because we wanted to isolate the effect of prior knowledge, we had a block design. So in the before block, we just presented 10 of those 10 2-tone stimuli in participants. Then we gave them prior knowledge by letting them watch kind of the templates that these stimuli were produced from, and as long as 10 distractors. And then in the after block, we just repeated the same 2-tone images as the before block. And in this way, the before and after block had the same presented participants with the same sensory evidence. They had the exact same 2-tone stimuli, but in the after block, participants had received prior knowledge. So in that way, we compared discrimination sensitivity between the before and the after block, and were able to test the effect of prior knowledge in isolation. And what we found, so was with dark green here is the before block, and with light green is the after block. All groups, so both age-matched controls, patients with Parkinson's, without hallucinations, and with hallucinations, they all improved as we would expect them to when they saw the prior knowledge. But hallucinators improved twice as much as non-hallucinators did. And this was also correlated with hallucination severity. So the more severe hallucinations people had, the more they improved after they received their prior knowledge. So this implies that in people who have Parkinson's disease and hallucinations, prior knowledge is relatively overweighted. So hopefully this is just kind of a summary slide to show you that hopefully I've convinced you that prior knowledge has a higher relative weighting in hallucinations and is proportional with severity. And this is important because, as I said before, we see the same kind of behavioral effect in psychiatric disease. But to try and figure out why this relative overweighting happened and where in the hierarchy this imbalance, computational imbalance may be happening, we then wanted to look at a white matter structure. And in addition, there's a lot of evidence that in Parkinson specifically, that white matter degeneration can be an early feature, both from animal models and from cell cultures. Axonal loss is seen much before grey matter loss in Parkinson's. So what we did is we used diffusion weighted imaging to assess white matter in vivo. And although a significant limitation of traditional analysis techniques, is in yellow here on the top, is their inability to model crossing fibers accurately, and that is 90% of voxels in the human brain, we used a newer kind of higher tensor model, specifically one called pixel-based analysis, the one that we use that overcome this limitation. And you can hopefully appreciate that on the bottom, you get a very more granular picture of where the white matter accents are in the brain. And we recruited 140 participants, of whom 107 had Parkinson's disease, and all of them had neuropsychology with a cognitive task per domain, motor assessments, as well as brain imaging at baseline, and then again at 18 months. And at whole brain white matter analysis corrected for age, gender, and total intracranial volume, already at baseline, we found reductions in fiber cross-section, so how thin the white matter fibers become in patients who have hallucinations, and particularly within the splenium of the corpus callosum and the left posterior thalamic radiation. And after 18 months, we saw additional extensive changes with over 30% reduction in many more primarily posterior tracts. Interestingly, hopefully you can appreciate it from the images, the frontal connections and a lot of the frontal occipital connections remain intact. And in contrast to white matter, grey matter remained relatively preserved, so there were no changes at baseline. And at 18-month follow-up, we saw just a very modest changes through a scattering of regions that you can see here in terms of cortical thickness. So what I showed you here is that there are significant macrostructural changes in the white matter associated with hallucinations in Parkinson's disease. It seems that posterior tracts are affected early, but frontal and frontal occipital connections are relatively preserved, and that may explain why we see this relatively overweighted prior knowledge behaviorally, because there is preservation of the connections that move the signal from the prefrontal cortex to areas further down the cortical hierarchy, and grey matter remains relatively intact. So next, we want to specifically assess the thalamus as a potential driver for both functional and computational network imbalance in hallucinations. And there's two main hypotheses that have been postulated in recent years about how the thalamus may be playing a key role in hallucinations. So first, the medial-dorsal thalamus is a key region that regulates PFC activity through both direct and indirect connections. And in schizophrenia, for example, where hallucinations are prominent, recent studies have shown both reduced volume of the medial-dorsal thalamus, but also medial-dorsal thalamus and prefrontal cortex connectivity. And a recent study by Eli Mitre and Glenda Halliday in Parkinson's disease using 24 post-mortem brains showed reduced volume and neuronal loss in the medial-dorsal thalamus in Parkinson's with hallucinations. Another hypothesis is that the posterior thalamus, specifically the pulvina, may play a key role in activating the default mode network. And there's a lot of evidence that the default mode network shows aberrant activity in association with hallucinations in Parkinson's. There's also some evidence that it may play a role in integrating sensory evidence with priors. So these were the two main hypotheses in terms of the thalamus as a driver for hallucinations in PD. And to test them, in the same cohort of patients that I showed you before, we used the recent automatic segmentation of the thalamus by Iglesias and others, and that segments the thalamus into 25 constituent subnuclei. And this segmentation was derived by using ex vivo MRI, histology, and manual segmentation of autopsy cases, combined with in vivo MR data from 39 subjects, so it was very robust. And so what we did is we assessed grey matter volume for each of the 25 thalamic subnuclei, but we also performed probabilistic stratigraphy from each of the 25 nuclei to the ipsilateral hemisphere and calculated the mean tract fiber cross section, which was the most sensitive metric from the whole brain study, and compared both grey matter volume and white matter tract integrity for each of these 25 subnuclei in people with and without hallucinations on baseline and after 18 months. And what we found was that first, looking at grey matter volume, there was no difference at baseline, but after 18 months, the right medial-dorsal medial thalamus showed significant volume loss in hallucinators in dark red here, compared to non-hallucinators in orange. And white matter connections from the medial-dorsal medial thalamus were also affected, with reduced tract integrity already seen at baseline, but becoming even more apparent after 18 months. And again, similar to what we found at whole brain, looking at the tract integrity of the right MDM, we found that longitudinal change was associated with higher hallucination severity. So thalamic tracts are affected in association with hallucinations in BD. The medial dorsal thalamus seems to be preferentially affected rather than the pulvinar or the posterior thalamus, and so both volume loss and connectivity loss of the thalamus with the medial dorsal thalamus with the prefrontal cortex may also play a role in an unregulated PFC activity in higher priors. And again, the generation of thalamic tracts correlated with the severity of hallucinations. So finally, just wanted to look at the brain network itself, so move further one level of the hierarchy further up from the level of the tract and the level of the region to looking at the whole brain network as a whole. And to do that, we kind of took a graph theoretical approach, and the principle when drawing a graph is that each region, for example, kind of different areas of the map in Kernenberg there, is a circle or node in a graph, and if there is a connection between them, for example, a bridge, then you draw a line to connect them. So instead of displaying these regions in terms of the physical distance, as in a normal map, we visualize them in terms of the connectivity. So we took we had structural and diffusion weighted imaging data and used them to construct a brain network graph for each of the participants. And I've just visualized this here as a connectivity matrix, and that gives you like immediately very clear information, I think. So each side of the matrix is a brain region, and the intersection between them is the strength of their connection. And with darker color, the stronger the connection between the two regions is. So you can see that immediately that brain regions are across hemispheres. So for example, frontal regions are very strongly interconnected across the two hemispheres, and also subcortical regions notated with S there are very strongly connected with all the regions across the brain. And then in the same cohort of patients, using the baseline data, we compared these brain network graphs across people with and without hallucinations. And what we found was that there was a subnetwork of reduced connectivity strength in patients who experience hallucinations. And this had 72 brain regions and 96 connections. And it's quite diffuse across the brain, as you can see, but it doesn't really give you a lot of kind of very useful information. But we wanted to see why these specific brain regions are affected in hallucinations, and also what impact would losing the connectivity between those regions have in terms of brain function. And one way to do that is using the framework of network control theory, which bridges kind of structure and function. And it has the main principle is that you view the brain as a collection of brain states, discrete brain states. So if this here on the left is a brain graph where each of the circles is a region in the brain and each of the lines represents an anatomical connection between regions, if you imagine that the white circles are inactive brain regions that are not firing and the purple ones are active, this can be perceived of as a brain state. And if, say, all of the brain regions were all firing and purple, then that is a different brain state. And using kind of a mathematical equation below, you can actually calculate all the possible brain states that the brain can go through using so the brain states are X, and you can calculate all of them over time T, but using just the structural connectivity matrix A, and the control nodes B, and the energy that you input into the system, and that's U. So just by the structural connectome and the energy, you can actually calculate all the different states that the brain can go through. And so if you imagine that you're kind of sitting in, you're in a state where your brain is kind of in the middle there, and you're sitting with your eyes closed and relaxing. But you need to move your brain into a state where you are drinking a cup of tea or you're watching a movie or something like that. Then you don't presumably need to activate a lot of brain regions to achieve that. So the energy required for you to make that transition is relatively low. But if you need to push your brain into a state where you're reading a book, then you need more regions to be active, so you need more energy, and even more so when you're playing chess, or listen to a talk, or you're doing something computationally more intensive. One metric that can calculate the importance of each brain region for switching the brain to all these different brain states is average controllability. And that is just a metric of how much important is one brain region for all these possible state transitions. So how important is the region for the brain to be able to transverse through different states? And what we did was, going back to that hallucination subnetwork of reduced connectivity strength, we actually compared controllability within the subnetwork across patients with hallucinations and without. And we found that patients who had hallucinations had reduced overall controllability within the subnetwork compared to non-hallucinators and controls. And also the regions that form the subnetwork of reduced connectivity strength, that lose connectivity in association with hallucinations, in our healthy controls, they normally have very high controllability. So they're usually regions that are important for state transitions. So it seems that the ability to switch between different functional states is affected in hallucinators. But more importantly, we wanted to see how this applies to specific changes in brain states. And I showed you way in the beginning that a lot of evidence recently from other studies that static functional connectivity is affected in hallucinations. So the default mode network is overactive, and there's increased connectivity between the default mode network and the visual cortex, etc. But although looking at functional connectivity averaged across an entire scan or an experimental condition is useful, in recent years there has been growing interest in studying time-resolved fluctuations in functional connectivity, because we know that regional changes in bold signal spontaneously fluctuating, representing this brain spontaneous fluctuation. So we need to there's been a move from this static functional connectivity to a more dynamic functional connectivity, where instead of looking at the average across the scan, you look at connectivity between regions at shorter time intervals and how these fluctuate during the scanning. And there is evidence that this dynamic functional connectivity is more biologically relevant, because fluctuations in FC over time correlate with EEG changes, for instance, across different bands, and also correlate with observed changes in brain metabolism assessed by cerebral blood flow. And also dynamic functional connectivity is relevant to human behavior and cognition, perhaps even more so than static functional connectivity. And this is just an example published a few years ago, showing that dynamic FC explains more behavioral variance than static FC, both on average across 58 behavioral measures here on the left, and in particular for measures relevant to cognition, such as fluid intelligence, working memory, etc., on the right. And this has been replicated in other studies as well. So dynamic functional connectivity is both biologically and behaviorally relevant. And so we wanted to look at specific changes that are seen in association with hallucinations. And in the same cohort, we looked at dynamic functional connectivity by applying sliding windows of 44 seconds, and then deriving functional connectivity matrix for each of those 44 seconds, and then assessing it for the duration of the scan. And then for each of these functional connectivity states per participant, we calculated the cartographic profile. So we calculated two measures: the within module Z score, which is the number of connections within a module that a region has, and the participation coefficient, which measures the distribution of a node's edges amongst communities of the graph. And if you have high participation coefficient, it implies that the region has a significant number of connections outside its own module. And so we use these two metrics to select the most integrated and the most segregated functional states, and then performed k-means clustering and allocated each of the functional connectivity states that we got from our sliding windows to either an integrated or a segregated state. And we compared the states between groups, but also estimated how much time each participant spent in each state, the dwell time, so the number of consecutive windows, and the number of transitions between those two states. And we didn't find any difference in the states themselves between hallucinators and non-hallucinators, using a wide variety of measures. But what we found was that patients who had hallucinations spent less time overall in this integrated state than hallucinators and control. So more time was spent in this segregated, less connected states. And this again correlated with hallucination severity using a hallucinations questionnaire. And the more severe hallucinations, the more time people spent in these segregated states. We also found that this was due to reduced number of transitions, and as you would expect, and it also increased dwell time. So once people entered the segregated state, they tend to stay there longer. But the most important question that we were interested in is, why do we see these dynamic functional connectivity state changes? So we know that there is pathology in the brain, so you have Lewy body and alpha-synuclein pathology leading to structural changes in both the white matter and the gray matter. But how do these structural changes lead to these changes in static and dynamic functional connectivity? And one way to address that is network control theory, but instead, so the same framework, but instead of trying to measure how much influence a brain region has overall, we use it to model the energy needed for specific state transitions. So for example, to go from the state here in the middle to playing chess. So you can actually model how much energy the brain needs to be applied in how many brain regions to achieve that transition. And so using again, our diffusion and structural data, we constructed a structural connection for each participant using the same postulation, and then you applied this framework to calculate transition energies, minimal control energies needed to transition from the integrated to the segregated state, from the segregated to the integrated state, but also the transition needed to maintain the integrated state and to maintain the segregated state. And we compared these minimal energies across groups. And what we found was that patients with hallucinations needed less energy to transition from the integrated to the segregated state than non-hallucinators and controls. And again, this was correlated to hallucination severity. So the more severe hallucinations people had, the less energy needed to achieve this transition. And this is important because it potentially explains why they end up, spending so much more time in this segregated state, because it's really easy to transition into that state compared to others. And then another benefit is that you can actually see which brain regions are more important into achieving that transition. So for that transition that we saw, there were significant changes in hallucinators, so integrated segregated states. We modeled how much each region contributed to the transition. And what we found was, so here, the darker the region, the darker red, are the regions that are more important for that transition. So you can hopefully convince yourselves that the more I haven't labeled the subcortical ones, but the subcortical ones and also the more transmodal regions of the default mode network in particular, were the main contributors to that transition. So they're more important into achieving that switch from the integrated to the segregated states. And finally, we wanted to see whether this regional contribution to this transition relates to specific neurotransmitters. So we know that neuromodulatory systems are important for transitions between functional states. And here's just an example of dopamine, specifically DRD2, which was amazing work done by Brown and others. And they used a working memory task and calculated the control energy needed to transition from a 0 back to 2 back state and vice versa. And what they've shown is that you need more energy to go from a 0 to 2 state than from a 2 to 0 state, which makes more sense because it's more computationally intensive. They showed that different brain regions contribute to this transition, and they also correlated genetic scores for DRD2 receptor in the prefrontal cortex, and showed that this was negatively predictive of control energy for these state transitions. And finally, they used amisulpride to an admission of amisulpride increased the control energy for transition, so they made the transition harder. So it's very convincing evidence that DRD2 expression kind of is associated with this state transition. But there's also other work from using other neurotransmitter systems. So Max Shine in 2015 did a drug manipulation study of the noradrenergic system, and Crinkleback recently showed the same in a simulation using serotonergic receptors. So we also wanted to see whether this specific transition is associated with a specific neurotransmitters pattern, and we used open access PET density maps for neurotransmitters in health for serotonin, dopamine, GABA receptors, and validated this using regional gene expression data for the same neurotransmitters from the Allen Human Brain Atlas. And these are the 3 neurotransmitters that we found significant differences in. So we found that 5-HT1B, 5-HT2A, and GABA regional distribution was significantly correlated with the distribution of how much regions contributed to the transition, essentially. So hopefully kind of convince you that there are widespread structural connectivity changes in hallucinations, and there is a subnetwork that seems to be very important for switching the brain between different states that is affected. Patients who have Parkinson's and hallucinations spend more time in a very segregated functional state, and this could be at least partially explained by the changes that we see in brain structure, because these lead to reduced energy costs to transition from the integrated to the segregated states. Transmodal and subcortical regions seems to be most important, and specific serotonergic and GABAergic receptors seem to play a key role. So hopefully I haven't talked too much, and I have convinced you that, you know, hallucinations are disorders of networks. They're not disorders of regions. Computational networks are affected, and there is an overweighting of prior knowledge, as well as differences in accumulation of sensory evidence. White matter connectivity is affected, particularly in posterior connections, with frontal and frontooccipital connections being relatively intact. The medial dorsal thalamus may play a key role by leading to unregulated PFC activity and overweighting of visual priors. We see changes at structural and functional networks at the whole brain level, with increased time in a segregated functional state, and reduced kind of ability of the brain to switch between states, and specific neurotransmitter systems seem to be involved. So I just wanted to thank everyone who kind of collaborated with all these different projects, all our participants for their time and energy, and you for your attention. And if you have any questions, I'm happy to take them.
Lovely. Thank you so much, and well done for having your talk ready without the slides.
It's difficult.
Yeah, I bet. I didn't want to be in your shoes, to be honest, but I think we got into a rhythm at the end.
Yeah.
Right. Are there any questions in the audience? Not yet. In that case, oh, yes, sorry, Valentina, go ahead.
Sorry, it took me a second to raise the hand. Amazing job. Thank you for the speaker, and also to you, Steph, because at the end, I was like, is she moving the slide now? Is she in control of the slide? Amazing. Amazing talk. I have a lot of questions, but I'm trying to regulate myself to leave the space for the others. So the first question, which is also related to one of the last topic you mentioned. If PD patients spend arrange their cognition according to the segregated state. Does this contribute to or impact other aspects of their behavior? And I think you were like kind of mentioning with the graph where you displayed the other systems that are involved in the segregated state. So I was curious to hear a bit more about it.
Yeah, no, that's an excellent question. I don't know for sure, but there's clear evidence that hallucinations are a predictor of a bad cognitive profile. So we know that people who develop hallucinations in PD are much more likely to develop dementia, and they tend to have a more cognitive phenotype clinically. So and less motor phenotype, if that makes sense. And there has been a really good work using dynamic functional connectivity by Furrer and Sasser and others in, I think it was Brain 2021 was her paper, 2020 maybe, and she showed that in PD with mild cognitive impairment and PD dementia, there was similarly a kind of more segregated brain state was more predominant. We took a lot of care to make sure that our cohort was quite well balanced, so there was no changes in terms of cognition on our patients with hallucinations compared to the non-hallucinators. So there were kind of MoCA scores, MMSE scores, as well as across the board. We did 2 cognitive tests per cognitive domain, and there were no significant differences other than one visual spatial organization test. But it's not but clinically we know that hallucinations are a precursor to more cognitive decline. So it's what time so the temporal kind of line is difficult to untangle, but I think that these are related, and there's a lot of evidence from other groups that these transitions are important for executive function, working memory, and stuff like that. So I think it's all interconnected, and it's difficult to disentangle. For sure.
And your study on the 2 different states was a resting state study.
It was resting state. It was resting state.
Okay.
Yeah. It would be great to have a task, but we haven't been able to do that.
Wow. There's always space for future directions.
Yes, definitely.
I'm gonna leave the floor to Lilith, but I'm coming back.
Yeah, sure.
Hello, thank you very much for the talk. It was super interesting. Just a question. What is the difference? I mean, I thought that patients with Lewy body dementia, they are like Parkinson disease dementia, but the only difference is that they have hallucinations. So what is the difference between Parkinson disease patients with hallucination and Lewy body dementia patients?
Oh gosh, that's inviting a lot of debate. So I'll try not to say anything too controversial, but it's this. There's a lot of debate, but actually it seems that more. So first of all, the definition for Parkinson's dementia, DLB, so the end phenotype is the same. So at the end, say 10 years down the line, it's very similar. So both PD dementia and DLB patients look the same, and they both have, most of them anyway, they have motor involvement, they have cognitive involvement, they have hallucinations, they have fluctuations. What we do clinically to differentiate between the two is that we say that if somebody presents with Parkinsonism, fluctuations, cognitive impairment, and hallucinations within a year, if they have cognitive impairment within a year of their motor symptoms, we label them as dementia with Lewy bodies. And if it's been more than a year, and then so they get Parkinsonism with or without hallucinations, doesn't matter, and then they develop cognitive decline, then we label that as Parkinson's disease dementia. So it is a little bit of an artificial definition, as you can appreciate. So I think it's the same, it's part of the same spectrum of the disease. At one end, you have a pure motor Parkinson patient who has a more motor phenotype, and then at the other end, you have the typical DLB patient who has, florid fluctuations, florid hallucinations, and cognitive impairment. Whereas each person is in the middle, it's more difficult to disentangle.
Yeah, okay. Yeah, thank you very much. And another question, kind of also on disease that are close to it, probably. So this one, like Huntington disease, like there are, as I understand, growing evidence that they also have visual problems, like atrophy in visual cortex, for example. So is there some maybe common mechanism why primarily kind of motor deficits, diseases, they also include visual problems?
Yeah, that's a good question. I mean, I think in terms of how visual hallucinations particularly are produced, I think that it's probably you need kind of multiple hits sort of in your kind of overall brain network to develop them. And it seems to me that you may have, a kind of a predisposition if your brain is wired in a specific way, and then once you develop kind of, I don't know, Parkinson's or Lewy body pathology, then that might tip you over the edge and develop hallucinations. Whether the link between other visual symptoms, because I'm not aware that I'm not sure that Huntington's disease patients have hallucinations per se. There is cortical spread, I guess, throughout the via synapses or with other. So whether there is a link between other motor disorders and the visual cortex, I'm not so sure. I think for hallucinations, there seems to be at least a lot of the behavioral things that and brain changes that people see in schizophrenia, we see in a more severe extent in hallucinations in Parkinson's. So I think there must be some link in the networks that have to be affected by a disease to produce hallucinations, and whether that is because of alpha-synuclein or whether it is because of developmental changes or whatever, they lead to the same end phenotype. And that's why it's important to look across disorders as well, I think, and study different conditions and but in the same symptom.
Yeah, thank you very much. Thank you.
I can interject here and carry that discussion further in the clinical domain. So you just talk about hallucinations in Parkinson's, but I was wondering what type of hallucinations those are, like visual, auditory, if they are very common across PD patients, or if it's just a few, if we know the differences, and roughly at what time they actually develop.
So they are quite well there's been a lot of cohorts, and it kind of gives different numbers, but they are common, they're not rare. So I think probably different studies give different numbers, but it's 30 to 70%. So I would say probably about 50% of patients will experience them with Parkinson's disease. They tend to be the very varied in terms of phenotype. So you have usually you have these minor hallucinations, so misperceptions. So a very typical thing is that people will see a pile of clothes on the floor and mistake it for a dog or a small animal or something like that. Or they can see shadows coming across the periphery of their vision. And then you tend to have these more complex, vivid visual scenes, typically faces, animals, people, very complex and very well formed. Visual hallucinations are by far the most common, but now there has been more studies that show that you can have auditory hallucinations and in association as well, or perceptions. So you can have somatic hallucinations as well. You can feel that somebody's touching you, but these are less common in Parkinson's. Visual hallucinations are by far the most common, and visual deficits as well. And it seems that there's something about visual hallucinations that it tends to be more predominant in neurodegeneration or in neurological disorders, whilst obviously in psychiatric disorders you usually get hallucinations in the auditory domain. And that's quite an interesting distinction, which we don't really have any answer necessarily for.
Yeah, absolutely. Valentina had more questions for you.
Yes, thank you. Because I was actually wondering the same thing that Steph and Lilith asked. But in terms of, the anatomical results that she presented has been presented in terms of loss, so loss of connections, like anatomical connection, loss of functional connection, loss of cognitive state. But is there room for plasticity induced by visual hallucinations, some sort of reorganization of the brain according to the new symptoms and new condition that arose? And this might explain also maybe hallucination induced by other type of syndromes or simply drugs, for instance. So not only a loss, but also enhancing at the same time other networks.
Yeah, no, definitely. And I think that is key. And that's why one of the most interesting findings that we saw in the white matter, like looking at white matter tracks, was actually that in hallucinators, the frontal connections and the frontal to occipital lobe connections were intact. And I think it's that combination that, so that you're losing one crucial connection, but you have the others maintained. And I think that other route allows for this kind of prior knowledge to kind of come downstream and influence your perception. And that may also partly explain why we sometimes see that in more, as cases become much more severe, kind of you lose insight, and people don't recognize that these are hallucinations, for example, or they might have more elaborate, like they might develop delusions or things like that. Whether the new functional connections are forming, or there's a reorganization of brain networks with a positive, in a positive sense, is really interesting. I don't think we have the answer to. We haven't found any changes in the other direction. So when we looked at structural connectivity and functional connectivity, we didn't find anything increasing, but work done by others has shown that the default mode network is way overactive, and the dorsal attentional network is also kind of underactive. And so there seems to be perhaps a compensatory change with increased activity in certain networks that should normally stay more dormant. So I'm sure that plays a role, but to what extent it's hard to say at the moment.
Yeah, as you were saying before, it's key to integrate findings from other syndromes and other studies, also with healthy participants, drug-stimulated healthy participants. And if I may, another question. Okay, thank you, Steph. Have you found any laterality in the in these patients?
In terms of the motor symptoms, you mean? Or in terms of our findings?
Your findings, yes.
Yes, there seems to be a right hemisphere predominance in our findings, both in terms of the well, the thalamic results, it was clear it was only the right mid-dorsal medial thalamus that was affected. But also, if you look at the kind of the sub-network, so if you just look at the network visually, the right hemisphere was slightly more affected than the left. We don't, that didn't seem to be associated with the motor onset side or the predominant affected side. And we don't really have a very good explanation for that. There was another group that also found right predominant brain changes. I think it was gray matter volume. And it is potentially interesting. I think an area of more research, to be honest. But yes, it does seem to be right predominant.
Yeah, and this is in line with studies also on anosognosia for Parkinson disorder symptoms in these patients, and some of the study report right lateralized association with right brain areas. That's why I was curious to see whether you would find the same pattern.
Yeah.
Thank you.
Thank you.
Thanks. I don't see any questions. I hope I'm not missing anyone. Thank you so much again. That was a fantastic talk and a great grand finale to our season of CNS Talks in 2022. Thanks for being flexible and letting me handle your slides.
Thank you for doing it. I don't know what's happening with my computer, to be honest.
You got the rest of the week to figure it out.
Yeah, fair enough.
But we made it. That's the most important thing.
Yeah.
Well, thank you so much for being here and presenting your work. Thank you all for joining, and I wish you an exciting week. See you next year, hopefully, for the new season of CNS Talks. Thank you all. Bye.
Bye.